Kabuki syndrome is a rare genetic disorder that affects multiple body systems, leading to growth delays, skeletal changes, and intellectual disability. Over 95% of all Kabuki cases are caused by the de novo loss-of-function mutation in either the KMT2D or KDM6A epigenetic regulators, referred to as Kabuki types 1 and 2, respectively. Although molecular causes of Kabuki types 1 and 2 are different, they share the same symptoms. Both Kabuki subtypes have characteristic DNA methylation signatures reflecting persistent chromatin dysregulation. Shared epigenetic landscape of Kabuki subtypes was never deeply studied, although it may reveal core regulatory pathways through which different genetic defects converge on a shared Kabuki syndrome phenotype. In this study, we analyzed two independent DNA methylation datasets representing the Kabuki types 1 and 2 patients and paired healthy controls. Whole blood of all studied individuals was epigenetically profiled with Illumina methylation epicarrays. As a result of differential methylation analysis, we have identified 49 hypermethylated and 65 hypermethylated genes shared between the two Kabuki subtypes. The hypermethylated genes were analyzed with Genome Enhancer pipeline to reconstruct the common disrupted regulatory circuits shared between the two Kabuki subtypes. Genome Enhancer pipeline is a constantly developed solution for automatized multi-omics data analysis. It first identifies genes characteristic of the studied pathology, then extracts their regulatory regions and performs transcription factor binding site search in their promoters to find candidate transcription factors and their combinations potentially regulating these genes. Next, network and pathway analysis is performed with the aim of finding potential master regulator that would be regulating a significant part of the disease genes via transcription factors acting on them. At the final step, where applicable, Genome Enhancer reports on the suitable therapeutic options acting on the identified keynotes that are serving as prospective drug targets. 65 hypermethylated genes shared between the two Kabuki subtypes were submitted to Genome Enhancer analysis. Promoters of these genes were scanned with a TRANSFAC library of positional weight matrices in order to find sites that are enriched in the promoters of the studied genes when compared to the control gene set. For statistical reasons, due to their rather short length of the binding site models, the site enrichment analysis finds lots of unreached sites. Genome Enhancer performs search for composite models of co-acting factors by applying a genetic algorithm that finds combinations of transcription factor binding sites that best separate the promoters of the studied gene set from the background control set. The constructed modules are further mapped to corresponding transcription factors, and consequent pathway analysis is performed from these proteins in order to find prospective master regulators. Here you see the transcription factors that were found to be regulating the hypermethylated genes shared between the two Kabuki subtypes. Network and pathway analysis is then performed starting from the transcription factors identified by the genetic algorithm. Instead of restricting itself with canonical pathways, Genome Enhancer extracts all reactions contained in the TRANSFAC database and uses them to find an optimal path from the selected transcription factors to potential master regulators capable of regulating a vast number of these transcription factors and underlying hypermethylated genes. As a result of this analysis, Genome Enhancer constructs a diagram that displays the connection between the hypermethylated genes with transcription factors regulating them and with master regulators responsible for the regulatory program of the studied pathology as a whole. Master regulators are represented by red rectangles on this diagram. Intermediate signaling molecules are displayed in green, and transcription factors are displayed in purple. Selected hypermethylated genes that are shared between the two Kabuki subtypes are displayed in light blue. You can see the ranked list of key master regulators that were found. These molecules are prospective drug targets that can be used in further investigations. The identified master regulators clearly point to the IGF-centered upstream regulatory program shared between the two Kabuki subtypes. This machinery is likely to be impaired in the Kabuki syndrome, reducing transmission of IGF-dependent developmental signals into stable transcriptional activation and causing coordinated hypermethylation and silencing of key developmental genes. The reconstructed molecular regulation mechanism connects epigenetic alterations shared in both Kabuki subtypes with the characteristic growth and developmental defects. With this, we have shown that the integrative approach of Genome Enhancer analysis enables transformation of DNA methylation data into a mechanistic framework, linking epigenetic alterations to transcription factor activity, signaling pathways, and candidate therapeutic targets, thereby facilitating interpretation of shared regulatory mechanisms underlying Kabuki subtypes. Thank you very much for your attention. Genome Enhancer is a constantly developed and updated pipeline available upon a license from GeneXplain. This work was funded by the European Union within the Chrome Rare project.